wabarakatuh and good morning everyone. As usual, my first question, can you hear me loudly?
Yes, doctor, you can hear you.
Alright, thank you. Okay, so before we continue, alright, to complete the topic of the PCR and sequencing by today, alright, so I would like to get some review on the Monday lecture. So what have you learned on Monday? Can you share with me a little bit? Oh, record first.
Recording in progress.
Can you share with me a little bit in the chat box? What have you learned on Monday? PCR process. Okay, more detail on the PCR process. We need. Okay, can you elaborate a little bit? What is the PCR process? More detail. What did you mean with more detail, Winnie? And then Ain, Nurul Ain. So elaborate a little bit. How about the rest? Hmm. How about the rest? To amplify a region or PCR cycle, Junhao said. Okay. Alright, you learn on the PCR cycle, you learn on the PCR process, you know to amplify a region. And then what else so anything that okay, the temperature needed for the process, and then what else? Anything that you can categorize it? Boleh tak kan category in group kan? What is the important things on the PCR that you need to remember? How many groups kalau you nak kategori, you nak groupkan? Komponen, component represent, or if it's reflecting to your lab report, it's under material and methods. Can you simplify using the material and method section? Kan? So what kind of material do you need to conduct a PCR? What kind of methods, okay, you need to have in the PCR? So first, I make it your life easier. Okay, what material you need? Material pun you can categorize kan, consumable, non-consumable, isn't it? So kita macam dalam primers, DNA, polymerase, DNTP, DNA template. So that one consider components kan. In the material methods, consider as kan kita nak buat lab work ni. Lab work ni report is very important, lab report. So under lab reports, you have to have introduction, objective, right? That is the basic lab report. Lab report ni actually is a starting point for you to write up your final year project thesis. It looks similar like that. Only that some element we put it. Okay, for example, hypothesis. Some of the lab report, if you still remember in biology kan, okay, in the chemistry, remember tak masa secondary school? Right, that's a hypothesis kan. Semua tu is actually reflecting your final year project write up, your thesis dissertation actually. And then second chapter is the material method. So I fall under material. Okay, you want in the objective, in the introduction, there's objective. Objective, I use Winnie punya statement, amplify a region, but that one is very general, very broad. Should be you specifically mention what is the region that you want to amplify, isn't it? Kan, you dah tahu dah PCR mesti spesifik. So, You must know, prior the experiment, you must know what do you want to amplify, right? Prior knowledge that with prior the experiments, you must know because you are designing the primer. Betul tak kan? So that is the objective lah, somewhere there. And then material methods. Okay, back to my question, material methods. Okay, so just to make it simpler for your life, under material methods, under materials, you can classify into what do you need to conduct the PCR? The components or the reagent. Betul tak? And the reagent, isn't it? So the reagent we need list out most of them: primers, DNA polymerase, the NTP, the DNA template. Any more? Any more? And the component or the reagent? Ada lagi tak? Siapa-siapa nak tambah? Tak cukup tu. Buffer, Junhao, thank you. And then, that one is reagent, kan? And then, what need you to prepare? Magnesium ion, correct? Okay. So the NTP includes the 4 nucleotide, right? The NTP represent all the 4. If you want to mention each one, then you can. So it will be apa? The NTP replace with apa? Mesti the NTP ni untuk semua. And the NTP is right? Equal to kalau kalau untuk A kita panggil DATP. Right? DTTP. DCTP and plus DG. Nucleotide for G, nucleotide for C, nucleotide for T, nucleotide for A. So when you read DNTP means it's included already all the 4. Jangan tertinggal, jangan ni pula. You left out this information. DNTP equal to all together. Okay, DATP, DTTP, DCTP and DGTP. You must put this one. Okay, usually we just mention the NTP, all the 4 nucleotides are there already. Alright, and then any equipment that you need, anything that you think that we should have before we should prepare to do this PCR? Reagent, and then we have consumable kan? Like consumable to consider reagent. Lagi, sometimes we call it raw material kan? Raw materials, right? Any equipment, special equipment that you need to have, you have to check first. Do you have this equipment in the laboratory or not? PCR tube, good. Mini, Sharifah, thermocycler, yes. Without PCR tube also you cannot do PCR. Okay, without thermocycler, that is very important. You can't do at all. Okay, PCR tube pun you tak boleh buat kalau tak ada. And because you don't have the appropriate case, appropriate box or appropriate materials, okay, to prepare your reagents. Remember, as a biomedical students, okay, you have to look at the appropriate apa ni kita panggil? Apparatus. Betul tak? Kan? Appropriate apparatus. So PCR tube for the PCR. Okay, PCR tube we have several volume. The smallest one 0.5 microliter. 0.5 microliter, very small one. Okay, the biggest one 1.5 ml, 1.5 ml. The biggest one. So usually we are using the smallest one, 0.5 microliter. Sorry, 0.5 microliter pula. 0.5 mil. 0.5 mil, 500 mil. Okay, ada kecil lagi ni. Until next week, okay, if there's no if it will be okay, then we are going to conduct the physical practical session next week. So I'm going to share with you all the kinds of the PCR tube and so on. So we are going to have some kind of practice. I will give you the things and then I will ask you to select the correct things that you need to do the PCR. So if you are selecting the wrong one, I will deduct your mark. Ada mark lah sikit as a lab report lah. Beside the lab report tu, I will consider the activity in the laboratory tu as part of that lab report. So next week get ready dah. Okay, don't panic. It's just to test either you understand or not, and then either you can think or not. You can think or not. Okay, so there is the things that, and then the other thing that you need to know, the methods, isn't it? The methods kan, what's first, what come first, what come next, and then principally you know PCR involved with the PCR process. PCR process means cycle of the PCR. PCR cycle, standard PCR cycle, 3 standard PCR cycle. Apa 3 standard PCR cycle? Saya tanya, saya jawab apa 3 standard PCR cycle? Anyone would like to give a try? Standard cycle. Begin with biasa in order lah. Biasa in order, denature, annealing and extension. Kan? Kadang-kadang extend, kadang-kadang kita panggil okay. So denaturation, annealing and extension. Thank you, Winnie. Okay, so that is the thing that we are talking last Monday. Kan? Last Monday. So we are talking more on the principle of the PCR. What do you need in the PCR generally, right? But supposedly in the practical session, you are going to look in detail all the raw materials that you need, the pipettes, okay, the pipetters, the tips tu kita untuk pipette, panggil pipette tip. Okay, pipette tips. Okay, we have here blue pipette tip. Tahu tak pasal tu? Blue tip kita panggil blue untuk big amounts, kan, yellow tip. Anyone know blue tips for how many amount? How many volume? How much volume that we can put by using blue tip? Anyone know about that? Ada tak yang tahu? Tips tu the sharp one, the sharp macam like needle that we can use that one to suck the liquid. Okay, so we have three different color, blue tip, yellow tip, white tip. So next week you're going to see that one. Okay, insyaAllah kan, if there is nothing yang I hope we can do that. Kan? Okay, so blue tip for big volume, yellow tip for the moderate volume, and then white tip, blue 500 watt. Leong, 500 watt. Boleh remember unit. Jangan lupa unit. Microliter. Okay, 500 microliter. Kalau 500 microliter berapa mil? 0.5 1 mil 1000 microliter, betul tak? Kan? 0.5 kan? Okay. So 1000 mil 1 liter kan? 1 liter banyaknya. Bukan 1000 mil, 1 mil equal to bukan equal to 1000 microliter. So 500 microliter, yes, you use, so 500 to 1000 microliter, you can use blue tip. Yellow tip up to 5,000, up to 500, 100 to 500, 100 to 500, yellow tip. Okay, sometimes yellow tip based on the pipetter juga. Kena tengok pipetter juga. Pipetter tu the one that we use to operate the measurement. Okay, next week we're going to see that one. Alright. And the white tip, you can use white tip for the smallest thing, smallest volume, 0.1 microliter. You nak ambil 0.1 microliter saja. Kecil ni. 0.1 microliter kadang-kadang. And then 0.1 microliter, susah nak cakap juga. 10 microliter. 0.1 mil, sorry. Okay. 10 microliter. Sometimes pun 0.1 pun nak ambil juga enzim. Enzim 5 microliter. This one. Usually 0.1 microliter to 10 to 20 we use white tip. Boleh? But still this one depends on the pipetter as well, because pipetter also different brand, different company, so they come up with a different range of the volume. They book in the pipetter itu maksudnya. So when you go to that particular lab, you're going to see. So there is an indicator there. Dia ada label kat situ. Okay, untuk big volume, you use this one. Untuk small volume, you use this one. Alright. Okay, now that is the material that you need to use when you want to do PCR, basic things. Okay. So next week I hope we can see that one. Alright, so today I want to share with you, continue with you the types of the PCR after you know that the standard PCR. Okay, this one we stop here, betul tak? Kita berhenti di sini, kan? Anything that you want to ask from slide number 1 until slide 42? Ada tak yang you nak tanya? Slide number 1 until slide 42. Anything that you want to ask me that you don't understand? No. As usual, no Semua dah faham? Okay, the one that we learned previously kan, okay, is more on the standard PCR. The result is like this. Okay, once again, the results is like this. After you put in the thermocycler, after 3 hours, you take out back the PCR tube and then you prepare this gel, agarose gel, and then you run. You put your PCR product here. Kat sini, masukkan dekat sini dengan together with the material that you can view the DNA. This DNA or this PCR product is colorless. You cannot see any colors. So without stain, you cannot see this PCR product. So how can you see this one? Because we are using stain, DNA stain or PCR product stain. Did you know any stain name? Tahu tak satu nama stain? Novel juice, bagus satu. Lagi? The conventional stain. Conventional stain before novel juice. Ethidium bromide. Ethidium bromide so scientists using ethidium bromide, which is so carcinogen, kan? If this ethidium bromide fall on your shoes, for example, you did not wear the full cover shoes, right? It will be harmful to you. It's a carcinogen agent for cancer. So ethidium bromide is harmful, so scientists develop a new material. Okay, novel juice. Novel juice is a name, actually, just a brand name. Okay, we also have Duret. In my laboratory now, they call it Duret. But the thing that you know is a DNA stain. You want to stain the DNA, you want to stain the PCR product. So when you order this to the company, you tell them, I want to stain the DNA to view under ultraviolet. Kan, kita nak tengok bawah UV. Kan, bawah UV, so we are going to use stain because our DNA or our PCR product is colorless. Colorless. Dia tak bercolor. Okay, unless you as the biomedical in future kan, you design this PCR product after 30 cycle, they change the color. They change the color, means the amplification successfully done. Kan, change the color, but the color is nothing lah kan, betul tak? Macam tu. So the strategy is the way that we can do to view this PCR product. Is there any amplification successfully done or not? So we can stain this PCR product. So after you stain, then you put in the well here. Masukkan dekat sini, and then you around 1 hour you you inject the electricity. Okay, then they will migrate. They will move from the bigger size to the smaller size. Itu prinsipal dia dalam gel electrophoresis. Okay, then you can see where the PCR product stick or remain there. So means that is your PCR product size. Itu maksudnya kan. So standard PCR at the beginning, mula-mula early standard PCR is qualitative. You just can see present, absent. This one absent. Absence of the PCR product. Not sure. Either you forgot to put DNA, okay, no DNA in this tube, or memang this individual doesn't have that gene, for example, that region, for example, so it won't amplified, isn't it? So many possibilities because the rest you got this result. So it's not so means that your experiment is correct. Maksudnya betul, tak ada apa masalah. Unless you forgot to put the DNA in this tube before you put in the thermocycler. It can be. It can be like that as well. Kan? Or the other factors is memang this individual doesn't have any region of this one. This one different from this one. So we can see the differences between individual, kan? Between the sample based on the length. This one based on the length. So this is one example of the standard PCR. Okay, it's a qualitative. We cannot we cannot calculate, we cannot count how many molecule DNA been amplified. Tak boleh, tak ada. Kita tak boleh tengok pun. Dia intensity, band intensity, right? Dia intensity lah. Kan? We don't know how many molecule DNA of you have after amplification. So this is qualitative. And then lower throughput lah because you cannot quantitate. And then in one gel, you can just run limited sample only because you are using your hands, right, manually. So that's considered lower throughput. If we can put in the equipment, 100 sample run together, for example, or 1,000 sample run together on the gel, then we consider high throughput. Barulah high throughut. This one consider lower throughput lagi. But still bagus, still great method. This one is still great method. Okay, for small laboratory. And then need post PCR step. This one lah, agarose gel electrophoresis. Means that after you done the PCR cycle, then you still need to do this to see the results. Kena buat ni untuk tengok results dia. After thermocycler saja you have nothing. You tak ada apa lagi. No result at all. Kan? Fikir. After thermocycler, no result yet. Tak ada lagi. So you have to do this, kan? And then higher contamination risk, as I said last Monday, it's not really there, or it's not really actually, because if you really you are not really clumsy ke, you are very apa kita panggil very good, your hands is so there's no shaking, kan? Then I don't think contamination can happen. And then several practice can avoid contamination ni. Dia tak ada lah, susah sikit sebenarnya. Specificity confirmed by size only. So limited jugalah because we have to know the size, okay, and then that's all the information that we can get. Okay, and then bands, however, bands can be further confirmed. So you can cut this, boleh potong because this is gel. Macam agar-agar, jelly you can cut. But don't eat. Don't eat. This one cannot be eat. Okay, so you cut this really at this one, and then there's a step that you can melt the gel and then just remain the PCR product in your tube. Then you can send for sequencing or do another step, insertion enzyme and so on. That is also you can do that. Further confirm if you want to do that. If the size, if you confirm by size it's not enough for you, you kata tak cukup lah, saya nak saya nak tahu sequence dia. I want to know this sequence, for example. Then you cut this one or you send PCR product in the tube. In the tube also can. Boleh? So now we go to the type of PCR. This is the list of the PCR type. Some of them. We have lots of the type of PCR, but this one quite common. Allele specific PCR, quantitative PCR ni short acronym dia can be qPCR. Q stand for quantitative, or sometimes they being called as a real-time PCR. Remember, real-time PCR, or sometime in the real-time PCR pun they put this one. Ada juga like this. Sometimes they use small letter, confused with the reverse transcriptase. So be careful. When you read the article, go back to the abbreviation. They refer and real-time PCR and the reverse transcriptase PCR is different one. So from the method, when you read the method carefully, you know. When they mention RT this one, for this one, you know this is real-time. if they mention RT for reverse transcriptase, from the method you know already. This one more to the conversion of the conversion RNA to cDNA kan. Okay, but because the reverse transcriptase tu acronym dia RT, small letters of the RT. Sometimes the real-time PCR also, some scientists use the small letters of the RT as well. So just for your information, this one boleh Okay. Okay. Alright. Then, so you know already reverse transcriptase PCR from the from the library last 2 weeks ago, right? Okay, and then ALICE specific quantitative PCR, and then we also have touchdown. Not only in the rugby have touchdown. Okay, rugby game. We in the PCR we also have touchdown. We also have nested. Another PCR type, multiplex, and then hot start. So in my laboratory, in my experiment for the postgraduate, okay, I always use multiplex PCR. Multiplex PCR. I very rarely, bukan very rare. This one where I never do this real-time PCR in my lab. Even RT-PCR pun di reverse transcriptase pun I very rare use this type of PCR. But this is a lot in my laboratory, in my experiments. Itu. Okay. The rest ALICE specific pun no. So because the one that I'm using, the type of PCR that I'm using is enough for me to get the result. So no need to do all of this. So all this type PCR is based on what is the purpose of the scientist. What is the purpose of you want to get? If you want to get this type of variation, then you can choose this one. This one, quantitative, means you want to know the amounts. Kan? You want to know the amounts. So you can do this one, real-time PCR. Reverse transcriptase is to you just want to convert the RNA to cDNA, kan? Okay. So this is the reverse transcriptase. Okay, reverse transcriptase PCR. The enzyme reverse transcriptase is used to make a DNA copy, a complementary DNA of an RNA template from a virus or from mRNA. For example, right, you want to investigate bacteria kan, or virus, right? Okay, the one that COVID-19, you are using PCR kan, COVID-19, apa dipanggil RT-PCR. So the one that you get probably RNA. You extract RNA from the virus, for example, right? Okay, then you have to convert it to the cDNA. So that we call it, during the conversion, we call it the reverse transcriptase PCR. Okay, type of the PCR that we use to convert first RNA to the cDNA. And then the rest, the cycle sama je. Depends on the laboratory, depends on the COVID-19 laboratory. It's not similar to the human, no. Okay, so this one, that's an example. For example, this one, you extract viral RNA, bacterial mRNA, protozoan poly A mRNA, ni. Kan? And then we cannot do PCR from the RNA sources. You have to reverse transcriptase RNA, this one, into the cDNA. Then you can do the PCR. Continue with the 2 primers. Nampak ni? Remember, kan, when you got a picture, go and carefully look and explain in detail. Easy untuk you all nanti. You dapat satu picture je, buat betul-betul tengok, and then you are not going to have any problem to explain on that. If you just read, then you have a little bit problem to understand and to memory that. This is what I'm talking from my experience. Okay, from this picture you mentioned here, any sources can get from viral RNA, bacterial mRNA, protozoan. Okay, this one, or from the human RNA, you can extract RNA as well from blood, blood sample. Okay, and then from this RNA, you have to reverse transcriptase everything, all the DNA sequence, all the RNA sequence into the cDNA sequence. Now you got double-stranded, kan? Double-stranded DNA. Then you can continue with the normal PCR with 2 primers. 2 primers, and that is real-time PCR. Now multiplex PCR. Multiplex mean, look at the name, multiplex, multi, more than 1 primer. More than 1 primer. Okay. Use of multiple set of primers to detect more than one organism or to detect multiple gene in one organism. Either you use multiple primers or you use one primer but multiple genes. Okay, multiple genes. For example, like what I did in my laboratory, I have 2 genes for one sample. So 4 primer. You ada pen tak sekarang? You tulis tak apa saya cakap? You jot down tak apa yang saya cakap? You dengar je ke? Yes. Winnie je ada pen. Or Winnie je using gadget. First years I forced them to find a pen and get anything that they have to write. Without lah, you cannot remember. Tolonglah, kan? As I can see from your answer. Daripada jawapan you all saya dah tahu dah ini memang tak tulis masa lecture. Kan? Sebabnya you lupa because some of my information is not there. It's not in the slide. Tak ada dalam slide pun. I remember during my time masa biomedic in the veterinary faculty. My time, my biomedic UPM still in the faculty of veterinary. Kan? During 1997. Kan? So that pathology lecturer, right? I really love how the way he teach. So I remember every single word he said, and then I transcribe similar exactly in the paper answer script. Macam tu. But it's not good. It's not good actually, right? Because you tend to forget everything without understanding. Kalau you dah faham, you memorize, then will be good lah. Okay, alright. Okay, so this one, use for example, E. coli, you have 2 genome from E. coli, from Salmonella, and then this different primer kan? This primer specifically for E. coli, this primer specifically for Salmonella. Okay, sorry. Okay, so you have 2 species, but you have same primer actually. This one same primer ni, same primer. Okay. But starting primer, remember, you short oligonucleotide only. So if Salmonella have complementary sequence to this primer, then it will amplify. It will amplify. But the results should be different from the E. coli lah. Either in terms of the length or in terms of the DNA sequence inside here. Kan? Kalau tengok, if you look at this picture, it look like it's different length. When we are using the same PCR primer, ini consider PCR primer sama, kan? Sama. So it will amplify at the different location lah, according to the complementary sequence to the primer. And then the other ways around multiplex to show you multiplex is like this. We have multiple genes, gene A, gene B, gene C in human DNA. We want to see 3 genes together. 3 sekali. Like what I did in my laboratory, GSTT gene, GSTM, and GSTP. All the glutathione gene, but glutathione is an island of the gene. There is several genes considered under GST, glutathione. So there are several. So we call it a cluster. And then 3 of them is the GSTM. So different primer lah kan? Different primer to recognize GSTM, gene A, to recognize GSTP, different primer, to recognize GSTT, okay, different primer. So all the primers are going to recognize different location, and then definitely they will produce different products. Okay, so and then in the human genome, I can see simultaneously one time. Okay, one experiment only, I can get 3 information, 3 gene already. And this one pula, one pair of primer, we can investigate on 2 different species. Boleh ke? Alright, okay. Okay, and then remember, there's a note here. Remember, the speciation is inherently biased depending on the GC content of the target and primer DNA. So, performing multiplex PCR can be tricky. So difficult to optimize. Remember tak? Last week, I told you, annealing temperature for primer, for every single primer is different. I can be different. So, how can we use multiple primers when they have annealing temperature different? Apa kita buat? Macam mana kita nak buat multiplex? What we have to do first? Any idea? My brilliant students dekat sini ni. Any idea tak? Kan? Takkan kita tiba-tiba terus guna kan? Betul tak? It's impossible, right? Okay, when we order the primer gene A, gene B, gene C, and then we immediately do multiplex PCR. Immediately do. But what we can do first? We do single PCR first, standard PCR first, and then we look at the annealing temperature, and then we optimize. For gene A, what is the temperature? For example, 60. Gene B, 62. Gene C, say 65, for example. And then give one try. All together, use gene A annealing temperature. Can you see that? That we call it optimization. Okay. Apa benefit multiplex PCR? Apa benefit multiplex PCR? From what we discussed, what I'm sharing with you just now, what's the benefit? What's the advantages? Can amplify multiple gene one time? Yes, you can shorten your time, right? Imagine you have 300 sample, kan? COVID-19, for example, how many strain? How many types of the COVID? Delta, Beta, Omicron. So can you imagine in one sample of the human, you can see all the strains of the virus in one sample, but different strain, different primer. Nampak tak? Kan? Yeah, boleh. So that is the advantages of the multiplex PCR. So we want to have this multiplex PCR to shorten our time. Kan, it's considered high throughput jugalah because you can see multiple genes at one time. Right. You remember basic genetics, okay? Not necessarily one gene is only responsible for that particular disease, isn't it? Probably multiple genes, polygenic, kan? Polygenic disease, disease that factors the multiple genes affected. So instead of looking at the one gene only, instead of amplifying one gene region only, you can amplify multiple gene. Okay. Okay, that's the advantages of doing multiplex PCR. Although it can be tricky lah to find the best annealing temperature actually for all the genes, for all the primers, for each gene. I remember kan, that's why you'll be introduced first with the annealing temperature and so on. Okay. That's multiplex PCR. Now we go to the semi-nested. Semi-nested, okay, remember all the type PCR, again, bear in your mind the purpose of doing reverse transcriptase because we want to have cDNA, kan? We want to have more stable DNA. And then multiplex, we want to multiply gene, we want to shorten the PCR and some more, kan? So semi-nested, why? Why? More specific and clean PCR product. Okay, last time I'm using nested PCR to clean my product. First primer that I'm using, it give me multiple bands. Supposedly one PCR product, one band only, kan? Very specific. So several times we already optimize the primer, we change the primer as already, and then still give multiple product, multiple band. Banyak. It's not specific. Right, okay, but so then I designing the second pair of the primer, like this, second pair of the primer, right? So this is first outer layer, then after you amplify, then you use a second pair of primer. And then not using DNA anymore, using the PCR product as your DNA target. Okay, to clean up. The reason that I'm using nested PCR last time during my time as a PhD student, okay, I'm using for the purpose of the cleaning product. I want to have a clean. Clean means I want one PCR product only, one lane, satu line saja. Okay, but this one show three primers are required. The normal upstream. Sometimes we call it upstream or downstream instead of forward, reverse, to indicate the different strand. And okay, as long as you know which one you refer. Okay, this one upstream primer, and then downstream primer, this one. and then as well as the third, internal primer. Internal considered as a semi-nested because 3 primers only. Kalau nested, complete nested means you do one time of the PCR first, and then you do the second round PCR. Right? But this one, in one tube, they put these 3 primer all together. The one that I'm using, different PCR. First, I have first cycle PCR for this red and yellow. After I got green one, this one, I'm conducting another cycle of the blue and yellow. Can you see that, the differences? Okay, so this just to give you idea why we want to do this. More specific, more clean product, kan? And then more to the pure product, kan? 2 rounds of PCR. Here dia guna 2 PCR juga. Okay, perform a normal PCR with the upstream and downstream primer, and then second round PCR with the downstream and internal primer. Either you want to the thing is you have to do the 2 PCR product because amplified first the big region. Okay, the big region. Second PCR usually smaller than the first one. Something like this. The second smaller product is the result of the second round of the PCR, and then a little bit smaller than the first PCR product. Boleh? So that is semi-nested PCR. And then this one, sometimes type of the PCR tu based on the name. The name tu the name tu come from the purpose. For example, like this, PCR for fingerprinting, DNA fingerprinting. Kan? Fingerprinting means we want to know the profile of your fingerprinting. PCR. So fall under PCR fingerprinting, it can be this list of the type of the PCR. And this list of type of the PCR. So just for your knowledge. And so AP PCR arbitrarily primed PCR. 1 primer required only. See, you don't have to remember everything. This one, a minor type of the PCR. What means minor means it happens on the certain laboratory only. Certain laboratory only. But this one globally, semi-nested globally, multiplex globally, common. Reverse transcriptase common. If you don't know this, means that I don't know. Probably I did not share with you the good knowledge lah. Kalau you tak tahu. If you don't know this one, still okay. Still okay this one. If you know this means that is indicate that you read a lot. You read a lot lah. Kalau you tahu yang ini. But if you don't know this one, something wrong with my teaching method, or something wrong with you. I don't know. Okay, I don't know. Okay, so this one for your knowledge, for your information, it can be the name of the type PCR too based on the purpose of doing the PCR. This one PCR fingerprinting, usually in the forensic kan, to investigate the suicide, to investigate the murder, the crime scene, the suspect, the natural disaster victims kan, the aeroplane punya disaster, right? So they use this one. They keep modify their standard PCR for fingerprinting. Okay, so this one lah, so I don't want to go through every single thing this one because it's not common. Tak perlu lah, just for your information. You know that we have other type of PCR lagi kan, but good to know, but not takut saya nak cakap not necessary. Nanti you pegang saya punya name. If you know this, people will know that you are very, you are really read on the things. You are a good reader. If you don't know this pun, nobody can blame you. Saya pun tak pernah guna this one. I don't really know. But when I prepare this slide, when I do the journal, the reading, I know this one. So I just want to share with you. Right, so ERIC PCR, enterobacterial repetitive intergenic consensus sequence. You know that this PCR targeting this one repetitive intergenic element in the enterobacterial, and then it's using 2 primer. Okay, itu maksud dia. So all of these fingerprinting techniques tell one if two isolate are the same or different. They do not provide information about the identity or relatedness of the organism. So this is the limitation still, but they can tell the difference, either the species are same or different. And then this one, this is the standard fingerprinting analysis either in human, kan commonly in human. Maksudnya ni sangat banyak. RFLP stand for restriction fragment length polymorphism kan. So maksudnya based on the different length among the individual. For example, we have two suspect or multiple suspect. Okay, multiple suspect. Kan in the crime scene. So you collect all the DNA from that multiple suspect, for example, then we see the differences in length after we amplify. That's we consider as the RFLP fingerprinting analysis. This one, the long name for this acronym, you know this already in the human genetic, kan? Principle of the RFLP, cutting DNA into fragment using one or a set of the distinction enzyme, means that if that individual have the site that recognized by the enzyme, it will cut. Itu principle dia. If your DNA sequence of that suspect DNA do not have the recognition site by the enzyme, it won't cut. Okay, remember variation in the individual, in the living things can be this single nucleotide polymorphism, isn't it? Kan? That is one variation type. Another variation type, duplication, repetition. Remember that one, don't limit yourself. Our DNA sequence, the variation can only occur at one single base pair. No. Kan, we learned this already, but you cannot apply this. Okay, the variation can be in terms of the duplication. Some of us has duplicate the region. Some of us only have one repeat. Faham okay. Tak faham tolong angkat tangan. Tolong stop saya. Okay, alright. So this one for chromosomal DNA, the RFLP fragment are separated by gel electrophoresis. So enough to see by using conventional electrophoresis gel, even though you said that is an old-fashioned method lah, doktor. Ayo, old-fashioned method, but it's very sensitive, very good sebenarnya. Kalau you kata, alah tak mau guna dah lah doktor gel ni. Apa? Old-fashioned lah doktor. Old-fashioned lah. Old-fashioned lah, but the thing is that one is the best. The best. Okay, separated by gel to transfer to a membrane. This one apa ni? Bila dah transfer to a membrane and probe with the gene probe, apa you rasa ni? Bloating, hybridization, right? This one dah bloating. Dia dah ada extra step. Transfer to a membrane and probe with the gene probe, hybridization, validation. See? The method that you use to screen the library, previously you also can use in the PCR. This PCR. After you run in the gel, transfer to a membrane, nitrocellulose membrane, and then probe lagi dengan gene probe. Nak validate. But for me, it's okay already when you can view the differences in the gel. Nampak dah perbezaan antara individu nombor 1, individu nombor 2. No need to transfer in the membrane anymore. Right? Okay. One advantage of this technique is that all bands are bright. Okay, because they are detected by the gene probe. This one, AP, PCR, ERIC, PCR, all have bands of variable brightness and also can have ghost band. This one mentioned talking about this method. Reverse back kat sini kan. Okay. Alright. And then for PCR product, a simple fragment pattern can be distinguished immediately on a gel. This one very fast method. Used to confirm the PCR product or to distinguish between different isolate based on the restriction cutting on the 16 S ribosomal DNA sequence called ribotyping. Also developed into a diversity measurement technique called TRFLP. So you just know the standard, you just know the principle, you know the DNA can be cut by enzyme, then everything you can develop. Many things that you can develop. So scientists, that's the wise of the scientists thinking when they know the structure of the DNA. When they know the DNA structure, we can cut. Oh, there's an enzyme, recognize the site, can cut because of that ability. So the techniques come, the type of PCR come. Another type of the PCR, DGGE analysis, they call it. Okay, denaturing gradient gel electrophoresis. Okay, ini nampak tak? Maksudnya dia buat selepas thermal cycler. Ini way to separate multiple PCR product of the same size. When you got very close size, kan? Okay, so you have to modify your gel electrophoresis. So that is also considered type of the PCR. Okay, DGGA use either a thermal or chemical denaturing gradient to separate bands on the basis of their GC content. This one, kan? When you use standard gel electrophoresis, it's not good to see the size. Then you can proceed with this one, DGGE, denature gradient. Okay. Alright, note of caution, PCR is inherently biased. Some primers work better with some target sequence than others, and primer will preferentially amplify target that are present in the high concentration. So scientists still don't know how accurately this type of analysis depicts the population actually present. Boleh juga. So some of the type of the PCR too, there's a limitation as well, and also there's advantages. Okay, this one TRFLP. Terminal restriction fragment length polymorphism. Yang tadilah kan? So a way to separate multiple PCR product of the same size juga. This product can be generated by using this one. And then perform as usual with the two primers, but one is fluorescently labeled. Different sikit. Okay, ada label dengan fluorescent. Then cut using restriction enzyme. RFLP, no, both primer are not labeled with any dye. This is what we call dye, fluorescent. So then, fluorescent label PCR please are detected. Right? Okay, this one. Okay, name. This is to show you. So when you are using labeling primer, you have to have the DNA analyzer. Our gel electrophoresis cannot detect the fluorescent. Tak boleh tengok. Okay, gel electrophoresis quite conventional, quite old, old school. Old school kita panggil old school method, but still powerful. So when you use primer, when you use anything label with the fluorescent, you need to have something to absorb the signal. Remember that one. Once you use fluorescent, you have to emit. Kan? You kena emit, absorb untuk tengok. Betul tak? Do you agree that? Physics punya principle. Once of chemistry, I don't know. Okay, once you use fluorescent, remember, you have to have another equipment and another machine. So that machine, we call it automated DNA analyzer, analyze the fluorescence. Analyze the fluorescence. So that's not the normal gel electrophoresis, what we call it, automated DNA analyzer electrophoresis. Okay, electrophorese juga, but not using the tank that we have in the laboratory. Okay, so that is the differences between that standard gel electrophoresis and this one, automated DNA analyzer. Okay, this one lah. This is the image of the TRFLP. When you label the primers, then you know the colors kan. But the thing is, if you use gel electrophoresis, you can just see the band, the size, but the fluorescent you cannot see. Kan, you cannot see. So you have to use DNA automated. You still can run in the gel, standard gel electrophoresis, but you just see the length. Okay, the length only. Nampak Boleh? Okay, now we go to the main. This one common. Quite common. Bukan not quite. Very common. Real-time PCR or the quantitative PCR. So you have to know this one. You have to know semi-nested. You have to know multiplex PCR. You have to know the RFLP. That one. Okay. So under fingerprinting just now, that small type of the PCR, not really lah. Okay. Ini apa beza dia? Allow quantitation of the DNA and RNA. Okay, because scientists now want to quantitate instead of just to see only the presence or absence of the product. So they want to count, they want to know the amount. Okay, so the name now is the real-time PCR. So reaction are characterized by the point in time during cycling when amplification of PCR is first detected, rather than the amount PCR product accumulated after a fixed number of cycles. Remember, go back to the graph. Cycle at the x-axis, y-axis is the amplification numbers, right? But that one is a standard graph for PCR. But for real-time PCR, so when you use this PCR, you can know which cycle is begin to amplify. Okay. Theoretically, we assume the first cycle will give you 2 product, betul tak? But the thing is, in the experiment, first cycle still no product actually. No amplification sebenarnya, tak ada. So scientists ni, they are using this one, the real-time PCR. The principle is, you know which cycle start amplification, then they will count from that. Itu yang, that's this statement come from. Okay, PCR product first detected during cycling. So instead of first cycling, second cycling, no. Then you are going to see after 10 cycling, 10 cycle of the PCR process, then you start see the graph naik, the graph increase. So means that at the 10 cycle yang ke-10 baru you dapat produk sebenarnya. Okay, rather than the amount of PCR product accumulated after a fixed number of cycle. Kan? So standard PCR you don't know about when it start amplified, you don't know. So that's why we have to introduce 25 to 30 cycle. Kan? We introduce more cycle because the first few cycle tu probably no amplification yet, nothing happened yet. It just kind of warming up the reagent, warming up the ingredient only, the cocktail only. Yeah, faham understand Okay, that is the real thing. Okay, but theoretically we assume one cycle will give you two product kan, two with the power of n lah kan. So the higher the starting copy number of the nucleic acid target, the sooner significant increase in the fluorescence observed. So real-time PCR use fluorescence. Okay. Alright. So why real-time PCR? Kenapa? Advantage of the real-time PCR inilah. This is the list of the real-time advantages. Can monitor real-time. Dia nak monitor real-time, apa you perlu ada? Extra equipment. Extra accessory. When you want to monitor the amplification real time, you nak tengok, what can you use? Boyce angle. Boyce angle, what can you use? What is the additional equipment that you need to have? Apa? Computer lah, right? Right. Now the computer is the best equipment, right? As a medium. We can see, we can watch the movie and so on. We can see the image. Betul ke? You agree with me? So you have to have computer. So to monitor everything. So in the real-time PCR, extra equipment, computer. And then when you use the fluorescent, you have to have absorber lah to emit. Kan? However, the advantage is this one. You can monitor it real time. Okay, and then wider dynamic range of up to this one so specific and 10 fold, taklah more, at least up to 1,000 fold, up to 1,000 fold. No post PCR processing after the product. So when you put your PCR tube in this machine, leave it there. You don't have to prepare the gel anymore because you can see the graph in the monitor. Computer monitor. So no post-PCR processing of product. That's a good thing. No gel-based analysis at the end of the PCR reaction. Okay, this one. And then ultra-rapid cycling. Within 30 minutes or 2 hours, we can get the results. This one, standard PCR also can have rapid result juga, but because we did post PCR processing, right? And then highly sequence specific. This one standard PCR pun macam tu jugalah sebenarnya. So this is the list of the advantage of the real-time PCR, and then not disadvantages lah. Okay, I will change this a little bit lah. I said limitation. Kita tukar sikit, kita taklah kata disadvantages nah, kita letak limitation. And barulah then will be good, better lah, and then consider it as a disadvantages. Okay. Dah save belum? Okay, so limitation still, every method's got their limitation. Require expensive equipment, a set of the computer, reagent as well. When you involve with the fluorescence, definitely is quite high, quite expensive. Okay, and then due to there is extremely high sensitivity, you may get high deviation ni pula, of the same experiment. So you have to use internal control gene as recommended. In gene expression experiment, particularly housekeeping gene, kena guna control ni. But in every experiment, bear in your mind, you have to use control jangan lupa nanti your result is not valid. Okay, kalau tak guna control, your result is not valid tak valid. Okay, when you design laboratory, bear in your mind, put positive control if possible, and then put negative control, right? So internal control gene is important for the real-time PCR. Internal control gene maksudnya dia normal genes, housekeeping gene we call it. Okay. So tak ada dalam slide saya ni. That's why you have to jot down. Okay. Just remind yourself, I'm coming today, I want to know, not because of the exam. I want to know, okay, what are the lecturers are going to share with me today? Something like a webinar, kan, talk about money, talk about motivation. I want to know, kan, I want to know what, so every time you come to the class, okay, prepare yourself, say to yourself, I want to know, not because I need to know, no, I want to know. Lain nanti you stress. Kalau you tak letak macam tu, you can stress. I want to know. Put in yourself, I want to know. I want to be more knowledgeable. Kalau I want to be more knowledgeable, sometimes it will pressure you. But when you said, I want to know this, for me, it's just kind of give you motivation that you are going to fit yourself kan with the knowledge. Ada beza Cuba you cakap sekarang. I want to know about this. Okay, so you don't have to force yourself to come to the class without interest. Tak ada interest Okay, so this is the limitation of the real-time PCR. Okay, and then this is the equipment. Looks similar to the standard PCR, right? But the thing is, kat sini there is some modification. Here there is some camera device, and then this one are not connected to the computer. This one not connected to the computer also. So you can see on the big screen here, big monitor here. Right, and this one, nothing can see. Okay, but actually they have to connect to the computer. This one lah. This is the full set. The computer, the what we call station, real-time PCR station. So if you don't have the computer, come together with the equipment, you have to buy it, extra cost. And nowadays they come with the things, the station and the computer. Why computer? We need the software to analyze the fluorescence and so on. Right, the fluorescence is only intensity, remember? Intensity kan? So you need the software. The software costs you lots. Software tu sebenarnya kan? So this is some of the equipment, real-time PCR, real-time thermal cycler. Inside this, the thermal cycler process. Kan? Only the differences is they have the probe and so on, and then connect to the computer and software. Right? So theory of the real-time PCR. Okay. This course SPP 3503. Rick wanted to have a more under real-time PCR because more laboratories, majority laboratories have this PCR type. So the rest to just introduce to you here. Once again, definition, real-time monitoring of the amplification reaction, and then the purpose is to estimate the initial quantity. Kita nak tahu initial quantity, then we can explain the rest. Okay, not the end. Okay, so this is real-time PCR. It depends on the labeling approach. Labeling approach mean inside the reagent, there's something that they use the probe. Beside the primers, we add another reagent, which is the probe. So this probe label with the fluorescent. So type of labeling, it can be cyber grade, it can be Taqman, it can be hybridization probe. Okay, Taqman fall under hydrolysis probe type, hybridization probe type, and then cyber grade, this one direct probe type. Okay. Okay, so this technique allow quantitation of the DNA and RNA. Okay, once again, lagi sekali dia cakap pasal initial product first detected. The higher the starting copy number of the nucleic acid target, the sooner significant increase of the fluorescence observed. Okay, in the principle of the qPCR or real-time PCR, we can group the information into three. In terms of the chemistry, in terms of detection, in terms of the analysis. So when you do the real-time PCR, when you do the qPCR, you have to consider these three. Okay, what is the chemistry involved? What is the chemical reagent involved? What is the chemistry reaction involved? And then what is the detection involved? And then what is the analysis that I need to have? Okay, in terms of the chemistry, because in this approach, we use fluorescent dye and probe. So because of that, it will establish a linear correlation between PCR product and fluorescent intensity. When you get the fluorescent signal, that is the PCR product. What this statement means, and then detect how we can detect this dye, and then monitor. So fluorescent detection to monitor amplification in the real time. Analysis from the software, we need the software to analyze and estimate the template concentration. And then detection in real-time PCR. Okay, so this is the under detection. Then under detection. Okay, divided into three type of probes. Okay, use fluorescent as a reporter by three general methods. Okay, hydrolysis probe. This is the name. Okay, when you read the journal or when you heard the lecturers mention, Taqman real-time PCR, so means that the probe that used had this method, hydrolysis. Hydrolysis mean digest, kan? Digest, it will digest. So based on the name pun dah tahu dah. Beacon digest, Scorpion digest. Hybridization probe means attach signal. Attached signal. Okay, most accurate and specific. This one, light cycler, the name. Cyber greens, DNA binding agent. They bind saja and then it give the colors. Less accuracy because of the some limitation. But this is the common. Sangat common, cheaper, easy. Ini easy. Okay, then reaction content between. Okay. Which one is real time? Reaction content of the real time, and which one is the standard PCR? Oh, okay, this is two set of the reaction content. Can you recognize which one is the standard PCR and which one is the real time PCR? Sebelah kiri apa, sebelah kanan apa? Sharifah, try right is the real time PCR. Dia berjanji pula dengan Winnie. A pair. And then Winnie pula answer on the left side. Bagus. Okay, good try. Yes. Sebelah kiri, kenapa sebelah kiri standard PCR and sebelah kanan real time PCR, Sharifah and Winnie? What is the differences? The presence of the cyber green. Yes, cyber green ni apa? Where's the cyber green ni? Primer or probe? Stain. Probe. Probe dia memang action dia macam stain tapi dia probe. Stain saja kan Wan. Be careful with the terminology ya. Stain is not really appropriate lah. Probe ni memang dia bawa complementary. Stain dia tak complement kan, stain saja, no specific. Boleh juga nak guna stain but no. Jangan ambil pula perkataan saya ni, boleh guna tapi tak boleh. Ada juga student yang guna cybergreen ni stain. No banyak kali saya dah mention dah, no it's not stain. The differences between probe and stain no specific, betul tak? Dye stain kan, dye tu kan dia tak spesifik pun, right? Ethidium bromide, novel juice, durite that we discussed earlier is staining because it's not specific. Probe is carry complementary, carry some sequence. Primer carry some sequence. Differences between probe and primer, tengok balik. Tengok balik. Saya discuss that one very detail kan. So the differences between real-time PCR and standard PCR is only the probe. Volume ni depends on your laboratory. Don't memorize it. But you can see that the primer is less volume. Sikit je. Compared to the buffer memang banyak. Milli-Q water is a buffer. Distilled pure water. Buffer is buffer. And then we top up with the water. And then tag polymerase because both amplification. Both. Okay. So how to measure the PCR product for real-time PCR? Sometimes they divided into directly and indirectly. So cybergreen, which is the DNA binding agent method, right, fall under direct. Direct kan. So the quality of the primer is very critical. Kalau you measure the PCR product directly, the quality of the primer is very critical. Indirectly, in addition to primer, add fluorescent label hybridization probe. Ada probe lagi. Okay, double. Indirectly macam ada double work, double step, double reagent. Okay, right. Okay, now in terms of the chemistry, tadi kita go detection first and then now we back to the chemistry. Back to the nature of the fluorescently label tu. Kan, kita tengok sedikit apa maksud the chemistry. So, qPCR plus chemistry. Okay, so based on this picture, right, this is you can match with the name. Nanti you guna your gadget, you guna your laptop, you annotate sendiri. This one match to what? Tagman, molecular beacons, Scorpion ke, ampliflow probes ke, cybergreens ke, boleh. You match it. Partner it. Ini apa dipanggil? Tagman yang mana? Which tagman? Which tagman? Which cybergreen? So this one. Ini macam hydrolysis kan? Nampak macam dia ngap. Ngap. Hydrolysis ngap. Makan. Hydrolysis lice kan? The lice. Okay, then this one cybergreen. Okay, fluorescent detection. Sorry, not cybergreen, not yet. So this is just to tell you, okay, fluorescent detection by using light. Because of the fluorescence too, it will produce the light. So this light can be absorbed, can be emitted. There is a excitation. This is the principle behind the fluorescent detection. Chemistry kan, chemistry story kan, chemistry background, right? So when there's a excitation, boom, excited. So you can see the light, and then you have to emit its emission. Okay, so this is the principle of the real-time PCR. Now we move to the cybergreen. What do you understand from this picture? Apa you faham dari picture ni? What can you say on this picture? Sharifah, Winnie, you want to give a try? One, Muhammad Daud. Siapa lagi ada kat sini? Only bind to double stranded. Good, Winnie. Kan nampak tak? From the picture lagi dah tahu dah. So masa denaturation, dia bind tak? The first cycle of the PCR process, denaturation, these fluorescents are floating saja kan? Floating. So it will bind after primer extension ke or after can you see that? Why is less accuracy? Kenapa dia less accurate? So remember previous slide you mentioned cybergreen direct measurement and then DNA binding agent. But then why is less accurate? Anyone want to give idea? Inside your PCR tube, right? Sometimes still there is an original double-stranded DNA kan? The one that never touch with the primer, kan? Boleh jadi kan? Betul tak? Am I correct? In your PCR tube, right? For example, you put 100, we consider 100 molecule of the DNA double-stranded. But then, actually in the tube, only 90 molecule. 90 molecule, okay, 100, sorry. 100 molecule. Okay, we put 5, cannot be banyak sangat. Kita letak 10 microliter of the 10 nanogram DNA. So that one we consider 10 molecule, 10 molecule of the double-stranded DNA original with the 3 billion, 6 billion that one. Kan deployed. Right. Now, okay. After 25 cycle, you produce million, betul, million. But then the thing is, there is one original DNA molecule probably are not been touched with any primer. Tak pernah sentuh pun. Kan, possibly. Winnie dah jawab. Yes. So, this dye, cybergreen, also can bind to this DNA. Betul tak? Isn't it? Also can bind to this original DNA, the 10 nanogram at the beginning. It's not bind to the one that already amplified by using primer. Faham ke tak? Kan? Okay, remember the primer that different strand, different color tu, original black color, original strand black color. Okay, and then imagine the one that amplified by using our primer, blue color. Katalah blue color kan? Alright, and then in the PCR tube still got some of the black color of DNA. Supposedly, this cybergreen only bind to the blue color strand. Kan? Use macam tu. Okay, you draw a tube, PCR tube. Inside that, there is a black color DNA. Jangan banyak sangat. Kan kita letak sikit je. And then after few cycle, you get black color and blue color more. Can you get what I'm talking now? Dapat tak? Kalau dapat, angkat tangan. Okay? Dapat Respon sikit, faham ke tak? Angkat tangan ke tak? Boleh lukis ke tak? Dapat go. Boleh lukis ke tak what I'm trying to say to you? Okay, you draw. Originally, you have a less number of the black DNA original. Kan? After that, because of the primer, we got new strand, blue color. But still at the end, after 30 cycle, in your PCR tube, there's still a mixture of the black color. Black color tak pernah gone. Kan? Black color tak pernah gone. Betul tak? Black color still some of them there. Mix lah. Right. Then suppose this cybergreen only bind to the blue DNA, the new one that been amplified. But the thing is, it also bind to the black one, so less accurate. Instead of reporting double-stranded new with the specific target primer, but now it's also somehow is include as well a little bit from the black DNA. Faham tak? Nuril faham tak? Thank you, Go. Okay. Imagine in the real experiment, still we got black one. Yeah, because cybergreen love to bind at the double-stranded. Whenever it looks double-stranded, bind, double-stranded, bind, double-stranded, bind. Okay, Maizatul Harnani. Tak pernah sebut nama ni. Narmita. Okay. Okay, so this is cybergreen. Direct, yes, very easy. This easy. Senang. Okay, usually in the many laboratories use this method of the detection cybergreen. After amplification kan. So then after that, so nampak tak PCR tube dia dalam plate. Ini kita panggil high throughput. In the plate, this one. This is a plate. Instead of single PCR tube, insyaAllah lah, I will show you this plate, PCR plate as well, and also in the single tube, right? So this is, sometimes we can put our cocktail here. So this one, 96 sample can, 364 plate also there, ada banyak type, okay? And then you put in the real-time thermal cycler like this. There is a casing of the plate here, then press it to the insert this, and then they will read amplified first, and then they will read emit the excitation of the light. Okay, so cybergreen is a dye which bind to double-stranded DNA but not to single-stranded DNA, and frequently used to monitor the synthesis of DNA during real-time PCR reaction. When it is bound to double-stranded, it will fluoresce very brightly. And okay, like this. Okay. And then now second, under hydrolysis, right? Under DNA binding agent, only one. Okay, cybergreen only. Under hydrolysis agent, okay, hydrolysis probe, okay, there are several types. Taqman, Beacons, and Scorpion. So Taqman probe principle rely on the nucleus activity 5' to 3', Taq polymerase, okay? To cleave dual label probe during hybridization to the complementary target sequence and fluorophore base detection. Consists of the fluorophore reporter and attach the 5' and then a quencher at the 3'. Kat sini reporter, kat sini quencher kita panggil. So the quencher molecule quench the fluorescence emitted by the reporter when excited by the cycler's light source via FRET. What's FRET? Fluorescent resonant energy transfer. As long as the reporter and the quencher are in proximity, quenching inhibits any fluorescent signal. This is the principle behind the Taqman probe. Okay, alright. So kita tengok gambar lagi senang. So belanya. Okay. Alright. Okay. So this is our double-stranded DNA, right? It's already amplified. So this is 5', 3'. Forward PCR primer, reverse PCR primer. So once primer attached and anneal, and then probe we design somewhere here. Remember, it carry the sequence as well. And then look at this probe, Taqman probe, fluorophore here, quencher here. Fluorophore carry the reporter kan? That's why reporter. Alright. So as long as they are still in the proximity, maksudnya close to each other, dia tak anneal lagi, this quencher will inhibit signal from this fluorophore. Dia tak akan, this fluorophore tak boleh signal lagi kalau dia masih berada dekat dengan quencher in Malay. And then polymerization continue with the kehadiran Taq polymerase and the NTP for until here, until this probe. What they do? This polymerization will separate the fluorophore from the quencher. Betul tak? Dia still macam dia makan. Hydrolysis, right? Hydrolyze. When this fluorophore been hydrolyzed, been separated from the quencher here, it will give signal. Means that one PCR product already produced based on the one fluorescent signal being released from the quencher here. Faham? Understand? So that is the tagman. This is a better view. Quencher dye, reporter dye. Reporter dye, fluorophore. It's bind here. But without hydrolyze, selagi polymerization tak sampai, tak ngap dia, tak digest dia, ni kan, it won't fluorize. Boleh. Okay. Denaturation and hybridization of the probe at the same time. Okay. And then extension of the primer and strand displacement of the probe. And then cleavage of the probe and fluorescence from the reporter dye. Fluorescence from reporter dye is directly proportional to the number of amplicon generated. Compared to the cyber green, specific, right? Because we put extra probe here, kat sini. Once polymerization occur, barulah fluorophore ni boleh release, kan? Macam cyber green tadi, it's not so specific, right? So this one, scientists improve from the cyber green, become like this, tagman. Tagman chemistry, like this. Okay. Right? Okay. So, this is sometime in the tagman, right? In the hydrolysis and hybridization probe, sometimes it will show the same principle, and so something like this. Okay, this is hydrolysis. This is hybridization. Okay, hybridization usually use dual probe system. Okay, when they meet, they fluoresce. This one hybridization. However, in the hydrolysis, single probe only, kan, single fluorescent label probe. Fluorescence, it depends on the hybridization status. Kalau ada hybridization, barulah fluorescent tu boleh naik, kan. Tapi ada hydrolysis juga kat sini. Macam tu, boleh. So dual probe, dia guna dua probe. Ada dua probe. Simple probe, single fluorescent label probe saja. Itu je. Okay. Right. Now. Tagman, just now, can you see the structure of the tagman? Kalau saya go back to this slide. Which one is tagman? Notate sedikit. Which one is a tagman? Boleh notate tak? Which one yang you rasa tagman? Yang itu. Okay, go. Thank you. Others? Before the binding. Saya sambung sikit. Sebelum dia bind to the DNA, what is the structure of tagman? Yang itu thank you Wan Muhammad Daud. Betul ke Wan Muhammad Daud? Ada lagi seorang Wan Muhammad, Wan lain. Ada lagi seorang, kan? Okay, yang ini yang go circle first ni, once dia only one. Okay. Only one. Okay. Alright, thank you, Winnie. Okay, so thank you. You matched that one already. Okay, so which one is beacon and which one is scorpion? You rasa? Good try. Nice. Scorpion? Okay, so you can do like that later when you know the information. Alright, so I go back to the scorpion. Molecular beacon. Okay. Tengok So this is molecular beacon, the structure of the probe. Saya kena clear sekejap. Okay, the structure of the probe. Just now the probe of the tagman linear, right? Linear linear lah. So it can any shape lah, kan? Linear, and then quencher and reporter or fluorophore. Right? Now molecular beacons, the structure of the probe. There is a complementary of the strand somewhere here between reporter and quencher. So it's like dia macam close. Dia close dulu. Before it's ready to bind, it close dulu. Protected first, the probe. Doesn't want to allow the probe ni main-main ngada-ngada pergi tempat lain dulu kan. Okay, close first, and then somewhere here is complementary to our DNA strand, amplified one, right? Okay, so after denaturation, this will be opened up, and then it will find the complementary here state one. Okay, so this is the structure of the molecular beacons probe. Like this. Okay, dia buka macam tu. Okay, when they are denatured, this reporter and quencher akan will be separated, and then allow the target region complement to the specific region, right? Okay, so this is fluorophore, quencher, and template. Then, so the differences between molecular beacons and TaqMan probe is just the structure of the probe before they bind to the template. Right? Okay, and then now this is a scorpion. The one yang go tanda tadi scorpion dia sebenarnya, betul tak scorpion dia ada ni separuh molecular beacon and the primer together. Primer together. Primer sekali. This is scorpion. Like that. Okay. Once again. You boleh replay banyak kali kan. So this is the structure of the molecular beacons kan. But then the scorpion because attached together to the primer. This is the primer. Okay, primer recognize the specific target, bring together the probe, and then denaturation. Kan? So that's why it become hydrolysis group probe detection because the polymerization too will hydrolyze the quencher from the reporter. From the quencher lah. Boleh? Okay. So comparison of the probe chemistry in terms of the picture like this. Okay. Show here this is polymerization kan. Right? Boleh Okay, so another picture, comparison between methods of fluorescence detection between DNA binding agent, cybergreen, and hydrolysis, and this one hybridization. Kan, hybridization like cycler, dual probe, one from this direction, another one from this. So when they meet, they are not hydrolyzed, but when they meet, they will introduce FRET. They transfer energy of the fluorescent too. So they excite the fluorescence. Okay, this one bind to the bind to the DNA double strand. This one, okay, hydrolysis as well, molecular beacon, and together with the scorpion. This like cycler hybridization. Probe hybridize, and then when meet, it will excite the FRET. Fluorescent energy transfer. Okay. So this is now we go to the analysis of the real-time. Real-time PCR is a kinetic, because we see the real-time amplification kan. Amplification associated with the fluorescence at each cycle during PCR. This is what I mean. After 2 cycles, 4 cycles, 6 cycles, nothing happened. Still zero product. This one increasing fluorescence. Okay. This one until 19 cycle, then we start observe some of the amplification. This one. So because of this, in the mathematics, we cannot use direct graph kan, direct measurement. So we have to convert to the log. Kena convert, kena guna graph yang lain kan. So they convert, that's why they become delta. Ni nampak ni delta sebenarnya, delta increase delta fluorescent, delta absorbent. Okay, reading. Okay, so you have to convert it to the log. Okay, so no gel-based analysis at the end of the PCR reaction because we know already this one. So from this calculation, from the initial product, we can at least predict after 34 cycle how many. Kan nampak tak? We can predict. Because of this prediction, we cannot use direct graph. Tak boleh gunalah, you have to convert it. Okay, computer based analysis of the cycle fluorescent time cost. Kena guna ni. Okay, this one. There is a requirement, you have to consider this as a baseline from actually your graph 0 is here, right? But in the real experiment, now 0 become here. Betul tak? So from here to here, we call it CT. Dia macam tu, CT threshold. Okay, threshold kat sini. So no template control. This one, we use this as a baseline in the analysis. Okay, right. So quantitative, because we can see, we can count the amount of the amplification. Very specific, varies with the chemistry detection just now. High throughput, okay, eliminate post-PCR step, reduce contamination risk, higher reagent cost. Various chemistry you can use, okay, this one, right, okay. Multiply capability in some chemistry. Sometimes we can multiply between Cybergreen and Techman, or Techman and Scorpion, Scorpion kan, we multiplex primer and probes, right, okay. Alright, so this is once again quantitative PCR. Okay, instead of from here you start seeing the amount, but actually somewhere here, somewhere here, somewhere here. This is standard. So means that limit of detection means from here. This is standard kan? Standard. But actually after this cycle, barulah you detect. Then your experiment detected. So you have to consider this as the first detection. Okay. So you have to plot, okay, a new graph threshold. Boleh? So from here to here, consider as the C threshold. Okay, cycle of threshold. Okay. So every different sample, different threshold, right? So that's why analysis is very tough in the real-time PCR. Okay, this is so in the graph of the real-time PCR, we have to consider several phases. In the standard PCR, no, there's no phase that we consider. So real-time PCR has 4 major phase. So means that kalau kita guna standard PCR, kita tak nampak, right? We cannot see the real phase, right? But when we use real-time PCR, oh, actually from here to here ni we consider linear ground phase. For example, macam warming up phase kan? Warming up phase lah. Until 16 cycle, barulah early exponential. And early exponential. Then after that, log linear phase. Then plateau lah. Plateau boleh. Right. So this is the explanation on every phase. In the linear ground phase, what happened? PCR is just begun, warming up. Fluorescent emission at each cycle has not yet risen above background. Belum lagi. Baseline fluorescent is calculated at this time. Okay. And then this one, CT, threshold cycle, where the first significant increase in the amount of PCR product correlates to the initial amount of the target template. Where we begin to see the initial fluorescent that represent the PCR product, that we call it CT. Right? So represent the starting copy number in the original template. Okay, alright, bawah pula. And then early exponential, PCR just began at the same time. The amount of fluorescence has reached a threshold where it is significantly higher than background, usually 10 times the standard deviation of the baseline. Okay. So this is the example of the software. Okay, example of the software only. Different company may have different software. Okay, it may have different software. So you can see different sample, kan? Different sample ni, different graph, different exponential, right? Boleh? Okay. So and then another method. So this one you have to convert it to the standard curve method. From the log, then you have to make it linear. Okay. So instead of kan, this one threshold cycle. Terbalik dah kan, cycle kat sini, copy number kat sini, right? Kan, right. So this is example analysis, standard curve method, and also the threshold method. Boleh? Okay. So this is to show you the differences between standard graph PCR and the real-time PCR. So real-time PCR, we can see the starting cycle that trigger the amplification. Right. So now we move to the application of the PCR in general, semua sekali. Indirectly, I did mention to you already, kan? Some of the application of the PCR tu kan, we want to see the individual differences, variation, but some of them are more specific. Okay. Classification of organism, genotyping, different length, we call it genotyping. Which version of allele? Wild type, okay, present, heterozygous ke? So that is the application of the PCR. Molecular archaeology, so this is the list of the application lah. This one. Okay. Or sometimes application can be divided into basic research and applied research. So basic research, for example, nak screen, you want to screen mutation. You want to see that drug discovery, classification organism, sama je macam tadi juga. So kalau apply, more to the genetic matching, detection of pathogen, nampak tak? Ni, COVID. And so PCR, application of PCR being used in the COVID-19 detection, for example. Prenatal diagnosis, we want to confirm, and for example, some of the abnormalities, molecular, in the not in the chromosome level, in the DNA level. Okay, PCR we use to detect mutation in the DNA level. Okay, DNA fingerprinting, application applied. Okay, sometimes being category into three: molecular identification, sequencing, genetic engineering. If you want to do sequencing, you need to do PCR as well. Ni project, all the field that use sequencing, bioinformatics, memang kena buat sequencing. Genomic cloning, sometimes sequencing. Human Genome Project, sequencing. Genetic engineering, you want to modify, you want to do gene expression study, you have to do PCR. Ini molecular only lah. And, okay, something like the example Example. Ini macam COVID, you boleh relate to the COVID. So DNA is unique for each single type organism. So for example, we want to detect COVID-19 virus and then isolate. Kan? Organism can be identified by using PCR. Lagi in detail. Different type of PCR. What is the step involved, and also what is what you can have after you do the PCR. Kan? Nampak tak kat sini sequencing PCR. Masih ada PCR sequencing reagent. Step dia macam ni. AFLP, amplified fragment length polymorphism. What is the step? Comparison. PFGE, pulse field gel electrophoresis. Remember DDGE last previous slide? So this is another electrophoresis as well, to separate the size juga. This one, pulse field gel electrophoresis. RFLP, nampak? See? Ini after PCR. You want to see you want to separate the size nicely, kan? BDGE, kan? Ini PCR. See? This one, PCR. See? Can you see? Alright. Detection of unknown mutation, tak tahu mutation apa. So this is your whole genomic DNA. And then do PCR. Okay. See. SSCP, single stranded. Ada bunyi pula kat sini. Kejut saya. Okay. Represent. Okay. Classification organism. Lambat dia ni. We want to relate the organism to each other. We want to see any similarities, and also we want to see any differences. So you have to do PCR. See? Okay. DNA. We got small DNA from all of this, and then we amplify. Another analogy. Okay, see this one. What? 8 sample, ladder or marker. M stand for marker. Okay, 100, 200, 300, 400, 500, 600, 7, 8, 9, 1000 and so on. We also have 2000 marker ada juga lagi. Kan? For some time 10 base pair, 20, 30, 40, 50, smaller. Right. Okay, so then you classify. After you get this, you do the phylogeny tree. Okay. Kalau tak ada band, no pathogen. If there is yes PCI, there is a band, the pathogen is present in the sample. Like this, right? Tuberculosis. Okay, right? Okay, the concentration of the tuberculosis, okay, with the decreasing sample, sensitivity, to show sensitivity. With the less DNA pun, we still can amplify the tuberculosis DNA. Okay, with the 10, okay, ni picogram. F apa? F apa? F stand for what you did. This is pico. Pico smaller than micro. Microgram. F apa? Ada yang tahu? Femto. Thank you Muhammad Daud. Femto the smallest ke ke? Femto. Ada lagi ke bawah dia? Saya rasa dia tak ada dah. Is there any smallest unit than femto? So femto ada dah tak boleh detect. Kan? Ini control. So 10 femtogram means that very small DNA of the tuberculosis present on that particular sample, and by using PCR still can be amplified. Boleh nampak. To show you is the powerful of the PCR. See, the inverse image. Okay. Short tandem repeat. Short tandem repeat 18, short tandem repeat 20. Okay. Prenatal diagnosis. Okay, this is apa? Anak dia kan? Siapa ni? Macam nak baca ni? Alamak, your senior kan? Saya tak tahulah. Dalam answer, your senior did mistake. Standard ke tak? Ini represent siapa? This box, square box represent for who? How to read the family pedigree? Male. This one? Can you change this one to be male, this one to be female? Boleh tak tukar? Boleh tak? Tak boleh. Jangan buat. No, Jessie kenapa ada? Surgery boleh. Bukan. Simbol. Maksud saya simbol. Sorry Winnie. Symbol. Can you change this symbol to become female and this one become male? Kenapa no, Leon? Yeah. This is standard. Your senior buat. Ya Allah. Jangan buat tau. Don't do this. Don't exchange. Don't exchange this one and then you stated. Memanglah you stated the square box represent female. No, it's not correct. This memang standardly for male. This one for female. So jangan buat Saya dah remind dah. Dalam jawab soalan exam. Nanti sebar-sebarkanlah kat sini you yang ambil forensik. Leon buat. Jangan buat. Kenapa pula you tukar? Lahai. Without dia tak pernah bagi tahu kan? So kita dah tahu dah. Kalau cross macam ni apa ni maksud dia? Apa maksud ni? What does it mean? Carrier. Good. What does it mean this one? 100% affected. Disease ya. I'm so surprised lah. Surprise sangat-sangat. Saya surprise sangat-sangat. Saya tak tahulah. Mereka saya ada salah mention. Kalau you remember lagi basic. Family pedigree siapa aja? Saya tak ajar directly. Maksudnya saya tak ajar you baca, betul tak? Saya rasa saya tak ajar baca betul-betul, but indirectly saya teach you male, female, male, complete. Kan? Seingat saya tak ada kelas saya ajar betul-betul. Maksudnya we focus on the how to read the family pedigree. Tak ada, but indirectly dalam analisis indirectly we come across, kan? So maksudnya saya kena dalam basic genetic, I have to stress out lah. Bahayanya, oh my god. If you go abroad, malunya. Aduh, malu cikgu dan anak-anaknya. Aduh, memang saya so surprise lah. Aduh. Okay, so jangan buatlah, jangan tukar-tukar. Sebab kita tak mention pun kat sini. We know square box is a male, round box is a female. Aduh, kalau ada line kat sini cross macam ni, dah die. Betul tak? Dah tak ada dah. Kan? Mana? Kalau, maksud dia apa? What does it mean? Kalau yang macam ni. Apa maksud ni kalau saya letak line kat sini? Dead, die. Anak lelaki die kan? Itu tak kisah. Left or right doesn't matter. But then, for children, the first one you draw this one, we consider the first. After this, second. After this, third. Boleh? After that, fourth. We read from that. From left to the right, we understand. The first square you draw, children children. The first children, second children, fourth children. Boleh? The eldest one here. Not the youngest kat sini, bukan. No. Ya? You nak letak left kat sini pun boleh. Itu tak ada kisah. Ayah dia, parents here. Either the female here, male here, okay, boleh lagi. No big indicator pun. No, tak ada. Okay? Think wisely. Kalau you interchange the symbol representative, means that, it's impossible, right? Kan? Boleh? Janganlah, tolonglah jangan buat. Sedih saya. Okay, so macam ni. Okay, so this is no negative control, right? This is the affected children, the affected son from this carrier. So means that although the both because it got both kan, dia bukan heterozygous. Nampak tak? This one not heterozygous. Homozygous. From this picture, you can explain many things. This is homozygous for, tolong tulis sikit version allele dia. Untuk orang ni, untuk ayah dia, mak dia. Tolong tulis. Can you write down version of allele, genotype? Apa genotype version dia nak tulis? Untuk orang ni, untuk orang ni, untuk orang ni. Cepat. Genotype for this one, affected child, affected son. Thank you, Winnie. Father? Father and mother, definitely heterozygous, right? Heterozygous because multiple band. Kan? One of the 644 contribute from the father. One of the 644 contribute from the mother. So both haplotype at this particular region, these children got this one. Father, homozygous apa? Homozygous pula. Father, heterozygous. Father and mother. Tak ada orang nak tulis. Six 4 4 4 4 4. 204. One of the haplotype 644. Kan, betul tak? Okay. Plus, okay, now, what type of PCR can we use for this analysis? We got different land, so what type of PCR? What type of PCR? Real-time PCR? Do you want to use real-time PCR for this simple analysis? No, right? No, you don't have to do real-time PCR. No, this is simple analysis, different land. So, what is the type of PCR? Type of PCR. Is it reverse transcriptase? Now, I move to the type of PCR tadi. Sekarang application. Allele specific. Okay, good try go, but not really correct. Allele mention only one position, kan? Multiply. Why multiply? Multiply different gene, different species. This one is not multiple gene. This one not multiple gene, Junhao. This is same gene, different land. This is not indicate different gene, ini bukan different gene. Yeah, standard PCR, but what type, Jessie? We're going to stop at 11:30, because this is practical session, kan? 12 kan? Okay. This is not different gene. They are on the same gene. Original land is this one, original. Tahu ke tak? Surrender. RFLP. This is RFLP. One of the haplotype here on this individual and father and mother, different version from this one. Okay. So this is one haplotype. Cuba you tambah 440 dengan 204, you dapat berapa? You dapat berapa? 440 tambah 204. You dapat 644 balik. 644 yang tak cut kan? 644 uncut. Uncut mean the sequence there's no recognition site of the rupanya. This come from one homologous chromosome. This one come from one homologous chromosome juga. But, for example, kat sini kan, around somewhere here, in this sequence, there is a recognition site by enzyme. Instead of C, for example, this one, T. This one apa ni? Ni apa ni? Genotype apa ni? Ya, cut fragment genotype. Okay, betul. Ini, then what we call it? Hetero ke homo? Hetero datang, hetero yang ni. Okey. Anak dia 644 644. Homo 644. So another version is Homo 444204 and Homo 444204. Faham tak kenapa? Ni kromosom ni. This is the chromosome. One of the chromosome carry this version juga. One of the chromosome carry this version juga. Homo lah. Dia hetero because ada 644 and 444204. Bukan dia hetero sebab dia ada dua band. It's not hetero because of these two band. You have to recognize first what is the uncut and cut. Nampak? Ni chromosome ni. Means that 644 uncut version. The first genotype. Okay, second genotype hetero maksudnya uncut plus cut. Betul tak? Cut plus cut together with the cut. So three genotype. First genotype uncut. Hetero uncut versus cut. Okay, boleh? So this is uncut. One of the haplotype homologous chromosome uncut. This one show one of the haplotype cut. Clear ke tak? Kalau tak clear, kena ni. Boleh? Clear. Faham dah sekarang? Contoh. Leong kan, Leong. How many chromosome you have? Chromosome number 1, Leong. Ada 2, ada 3, ada 4. How many homologous chromosome, Leong? You have chromosome number 1. 1, 2, 3, 4, 5, 6, 7. 2. Kenapa ada question? Kenapa ada question mark? You don't confident. Setiap orang ada 2. One from your mother, one from your father, right? One from your mother, you don't know which version like, cut version or uncut version. Right? Correct? Until you do this. Until you do this. Kan? Faham tak? So when you do this, you can see that it will give you information on the which chromosome. This is how. Boleh? Namanya lah kat sini. Okay, sebelah dah. Okay, so you can get sample from chronic villus or amniotic fluid to investigate. For example, this one Down syndrome. Down syndrome, check for chromosome 21 gene, the presence of the chromosome 21 gene, for example. Kan? Down syndrome, no need to do molecular, we can use chromosome study because it's a trisomy 21. But for example, Angelman, Prader-Willi, you cannot see really on the chromosome. So you do PCR, get the father DNA, mother DNA, and then amplify the region on the Angelman or Prader-Willi. Kat mana? Kat mana region you nak amplify? That's why basic genetic, human genetic 3503 related. Sangat related sebenarnya. Kan? So you tahu dah kat mana micro Angelman Prader-Willi kat mana? Angel 1 micro deletion apa? Kromosom berapa? 15 kan? Micro deletion, sangat kecil deletion dia. So you amplify on that particular region je, sikit. You tengok siapa yang carry, siapa yang contribute to the children. Boleh? Boleh. Okay. Okey, ni lain application PCR selain daripada tu you nak detect Neisseria gonorrhea ni. Biasanya macam ni lah, kehadiran presence of the bacteria, presence of the virus, this one. HIV, okay, this is some information. Okay, asymptomatic, right? So for this chlamydia, for example, common sexually transmitted diseases. Okay, so what we can do is we get the sample from the host individual, and then we try to amplify the bacteria species. This one. And then this also use PCR, forensic testing, and many others. Okay, ni fingerprinting. Our fingerprint. Okay, they use PCR. So the DNA sequence used is a short repeating pattern. Inside our DNA sequence, there is a special location. Ni repeated pattern ni. You know this already in the human genetic Okay, this repeated pattern ni we use this because different individual, different length. So one set of the variable number tandem repeat locus are inherited from the mother and one set from the father. Sama juga. Probably just now, okay, from father 2 repeated only, from mother 5 repeated. So you can see the different length. Okay, itu. This one. Banyaknya. This. Can you go and see this one carefully? Boleh tak? Kalau dalam exam, tak tahu nak elaborate, tak tahu nak explain. Ni, individual A, dia carry one homologous chromosome dia panjang, long. Another one, short. Kan? This one individual B, individual C. See? Okay. Tertruncated kat ni gambar ni. Saya check semula saya rasa ni daripada forensic crime scene ni. Ini from suspected, from the DNA fraction get from the crime scene location rasanya. So they compare between individual C who match to this one. Let's say katalah forensic punya this one. Which one is matching to the forensic? Yang mana matching? Yang mana matching? Individual A, individual B, individual C. Go say B. B because present of the length, same length, right? So we can exclude A and C. Ni multiple. This one multiply. Multiply because one, tengok kalau kita guna yang purple saja. Purple saja. A and B got it. Betul tak? So we still cannot exclude A. Betul? So we have to have more location. Add more location. In forensic, we have multiple location of the analysis. See, kalau kita ambil green kan? All of them present. Kan? So we still cannot exclude the suspect. We still cannot exclude. We still cannot find the suspect. Right? If you are using this one only. Then if you are using this one, only B got it. Some of the allele are matching, really match. Kan? Nampak tak? Kan? See, if we are using only one, it's not enough. So we use multiple allele location. VNTR number 1, VNTR number 2, VNTR number 3. So we can expect, we can get B is the culprit. Please anak-anak sekalian, boys and girls, every single image in this slide, please tolong tengok betul-betul. Conclusion for the PCR saja. Okay, baik. Boleh sambung lagi tak? Kita break 5 minit dan kita tengok sequencing sikit. Boleh? Sequencing nanti kita buat practical dalam next week. Kan? 5 minit break I can now who steal my biscuit. I can know who steal my biscuit now. You do the genotyping, Leon. You get the DNA. How can you know, Leon? You have so many friend, kan? Friend ramai kan? Yelah guna PCR tapi macam mana you nak pick up suspect? You have so many friends. Kan ramai friend ni. Dalam kat sini ada 50 orang tu. Kan? Okay, kita break sekejap 5 minit. Biar saya tarik nafas sekejap. Alright. Tak maulah 5 minit. 11. 12 kita start 11:12. 1 minit je lah, 11:10 saya start. Cookies time. Okay. Sementara tu ada yang nak tanya? Boleh tanya? You punya junior, ramai yang tanya, suka tanya. Soalan survey. Okey, kita sambung ya. Ready? Boleh ke? Boleh ke? Ready? Okay, sekarang kita dah dapat produk. Now, we have the product already, kan? Like this. Ni yang tadi ni lah. Right, this one. So, some of the researcher want to know the sequence ni. Want to know the sequence. Is it correct this sequence represent this sequence? Ni nak tengok juga, betul ke kan? So, sequence means we want to know the arrangement of the nucleotide between this, inside this. That's we call it sequence, sequencing. So you have to define what is sequencing, why we want to do that, where and how. Indirectly saya dah answer dah tadi, what, why. Kenapa? Because we want to know the order, the arrangement of the nucleotide, and then after that we can do the further investigation. Either you want to replace the gene, for example, kan? After you know the full sequence, where is it the location of the mutation? Can we replace, for example? Kan? What is that? Sequencing means we want to read the full order, the full arrangement. Where is happen? How? Kat mana? Where? We can do on DNA, we can do on RNA, we can do on protein sequence, RNA sequence, DNA sequence. If DNA sequence means you read on the DNA. If RNA lah. You are going to see the uracil. If protein, amino acid. And then how we can do that? Macam mana kita nak buat? Okay, so illustrate same thing, you should be able to illustrate the sequencing reaction. And then analyze the sequencing rate. Analyze sequencing rate ni, I will continue in detail next week during the practical session. I will continue from PCR until sequencing. Okay, when you can produce the reading, that means that you are able to read the sequencing reading. Okay, so because of this, remember, because of this, sequencing the nucleotide DNTP modified to the DDNTP, dideoxynucleotide. What is the differences between DNTP and DDNTP? So when it's been modified to the DNTP, no polymerization after DIN has been added. Means that structure of the NTP, there is apa tu kat mana? 3 hydroxyl ke? Right, because of the 3 hydroxyl polymerization extension, right? Extension to the new building block can be add up. However, in this structure, the polymerization will stop. It cannot extend anymore. So because of this, it will produce fragment of DNA differing only by one nucleotide are generated. Remember, purpose of the sequencing, we want to read every single letter of that 4 nucleotide. We want to see the first one, which one? ACTG. The second is Y. The third is what? So that's why scientists modified this one because we add up. Then the NTP polymerization in the PCR, you add up the NTP kan? Am I correct? We add up a new one, complement the NTP. Complement the NTP. Right? In every single nucleotide. In sequencing, no, we want to read. Then, if you want to read that one, you cannot allow polymerization happen. So once this bind, it will stop. Okay? And then to read that, remember, to read the sequence, we have to label. If we just amplify and do the process without labeling, we cannot read. Remember colorless? Colorless, right? Kan? So all the nucleotides are either radioactive or fluorescent label. Kena label lah ni. Kena label. Different color, different nucleotide, different color, different fluorescent color. Alright, radioactive or in the old days. Now with this fluorescent, radioactive also carcinogen, right? Harmful. So scientists can replace now already with the fluorescent. Right? So sequencing, cycle sequencing PCR. Okay. Remember the previous slide of the application? In the sequencing, there is a PCR. PCR as well. Okay. First PCR, we amplified more first. Second PCR of the sequencing, this one. So means that you have to have standard PCR first. Okay, and then after that, second PCR, okay, you are using only for first strand sequencing. This one. Different a little bit from the standard PCR when PCR driven DNA sequence procedure non-exponential. Dia tidak menaik. Kenapa tak nak menaik? Sebab kita tak nak amplify lagi. We want to read only. We don't want to amplify anymore, the PCR product. We just want to read. So this PCR use first strand only. One primer only. And then dideoxy sequencing method. This is the first sequencing method being introduced. Fluorescent indicators. So this is the principle. Still PCR driven DNA sequence procedure. Mesti ada PCR juga, but then it's non-exponential. Okay, non-exponential. And then must be a labeling. Must be a label. Okay, so this is the analogy. The picture. Alright. So single strand PCR together with the DNTP. This is the DNTP base either A, T, G or C with the template plus primer. One primer only. Nampak tak? One primer only. Ini single strand PCR untuk sequencing. Dengar ni. Saya tekankan kenapa? Selalu student buat salah. Kan? PCR, we need, untuk buat sequencing, we need to amplify first. We need to have more PCR product. So you amplify by using standard PCR first. And then second PCR, we call it single strand PCR. When you only use one primer. The rest sama. However, you have another extra DNTP. DNTP. Together with the DNTP, you also have DNTP. And okay. Okay. Tadi, this one with the presence of the NTP only. So primer attach, and continue polymerization. Continue because no DNTP, right? Now with the DNTP present, this is the primer. Okay. Tak ada pula sambungan dia. Okay, so for example, the DNTP complement to here DT, DA, DC, and then suddenly come DDT. So it will stop here. Nampak tak? Boleh tak? Can you see? First ini DT, DA, DC. Sini suddenly come DDT. What happened? Is there any polymerization? Masih berlaku ke tak? Bila DDT attached to this one, masih berlaku ke tidak? Continue or not the polymerization? No, because instead of DD is carried out. So, what is the structure? No, 3-hydroxyl. Tak ada ni. Nampak tak sini? Can you see? There's no structure of this 3-hydroxyl. So no this one, no polymerization. DNTP only have this one. DNTP doesn't have 3-hydroxyl prime. Tak ada. So because there's no this one, okay, no polymerization. Faham tak? So kat sini we know that here is A. Is A for this strand. Okay. So another strand, continue. Clear? So that's the principle of the sequencing. Okay. So in one tube, okay, you put everything, the primer, just now this one, this and this all together in one tube, but different fluorescent tag. For C, orange color, blue color, green color, different color. Okay, plus a preponderance of DNTP lah. Okay, we still allow amplification. Okay, polymerization. Okay, allow juga, kena, have to. And then together with that, this. Right, so this one, this is another example. Primer sequence. Okay, and then this is the amplification. Kan, amplification, and then suddenly DNTP bind here. So stop. Alright, stop. Okay, so the excitation emission happen. Okay, berlaku. So there is a camera device to absorb this fluorescent color. So this is what color? Orange color, magenta, kan? And then the first one you read first, and then second stop here as well. Remember, this is PCR product, kan? You already done with the first PCR. So what you get here, you use PCR product is specific, short only, very short, kan? Short product already. You amplify, for example, 200 base pair. 200 base pair in the first PCR. First PCR. So this one, you use this one, you take 1 microliter, put in the tube. So inside that tube for sequencing, there is a 200 base pair DNA, not the longest one. Faham tak? And then you want to read 200 base pair nucleotide. So imagine this is 200. The longest one, right? The longest one. This is 199, position 199. Okay, so they will read like this. Clear? So this is the first method used by DiDoC. Okay. Okay. the idea is you give different color to the different nucleotide, so you can read that. Okay. So this one according to the represent the length, the location. Shortest 5 prime. Nampak tak bacaan dia? Longest at the 3 prime. Because our primer attached to the strand, right? Sorry, our primer, yeah, our primer attached to the strand. Okay, so we read. Okay, the longest is 3 prime. Remember, the shortest is the 5 prime. However, in the sequence, then when we use the software, it will translate like this in terms of the peak. Into the peak. So we read based on the color. Orange color is A, kan? Green color is T. Okay, C. Sorry, yellow, orange kot. Boleh lah. Tapi different color. Okay, so this one, this is what we call it, sequencing reading. Ini dalam gel. Correct. So this one. Okay, so this one 3 prime to 5 prime. Terbalik sikit kan? Betul tak? Terbalik. Okay. So then you convert it to become 5 prime to 3 prime. Boleh? Dalam reading, inside the reading, you read from, you not read the order 3 prime to 5 prime. Right? In the gel also, longest to shortest, 3 prime to 5 prime. But we have to read from the 5 prime to 3 prime, right? So to read this sequence, convert it back. 5 prime, 3 prime. Jangan ambil 5 prime, terbalikkan. Don't terbalikkan, don't reverse. But find the complement. Find the complement. The complement for this one, 3 prime, is TTAGA. This is the correct answer. Sorry. Okay? Okay, ni. Contoh. Example again. Okay. Right. Okay. Like this. So this is a classical without labeling. So we just read based on the different length. This with PCR. So when different tube. Ni different tube. Because of we know that we can label the nucleotide with the same, with the different fluorescence, so we can prepare one tube only. But in the classical sequencing gel, this is different tube. One tube for G, one tube for A, one tube for T, one tube for C. So you have to prepare 4 tubes for one individual. Then when you run the gel, you're going to sort this one. Inside that particular G, kan? Because it's only have DDG, so it will show you only C sequence. This C kan? Kalau dia guna G lah, G for DDG. Means that ini C. Kalau ini adalah bacaan G, maksudnya dia akan guna DC. Faham ke tak? Tiup ni mesti contain DDC. This tube mesti contain DDT. Understand? Respond sikit, understand or not? Inside the tube, okay, ini. Inside the tube. To investigate G, you have to put DDC, correct? Correct ke tak? Kan? Because C complement to G. Okay. Sharifah boleh, Sharifah? Go. Leong tadi, 1. So if you want to investigate A, you put DDT. Okay, right. So clear ne? So this is classical without fluorescent label, but with the fluorescent label like this. Now, remember, as I said again, when you use fluorescent, you have to have something like this. Then you can read. Means that you have to have monitor, you have to have software. Itulah maksudnya. Okay, but better than this one. Susah kan ni? Hard work. Reading classical sequencing gel. Inilah tadi. Ni classical sequencing. Boleh tak tulis kat sini? Siapa nak tulis? 3 prime. Kat sini 3 prime ke 5 prime? Dia mula pada sini lah. Longest, shortest. Longest, shortest. Longest 3 prime, 5 prime. Begin with the, start with apa ni? Start dengan apa ni? What letters? What nucleotide come first? From left to the right. Bottom, top to the bottom. A, second. Second, C. Third, A. Kan? Third is A. Susah kan? Ni. Fourth, C. Fifth, A. See? Boleh tak nampak? So, from this classical sequencing gel, we read like that. But compared to the modern one, like this. Which one is easy? Which one is easy? Modern? Modern one. Modern one easy lah. Okay, so this one. This one is modern. Okay, in summary, wherever you got DNA sources, you can do PCR and then you can amplify your desired region. Okay, then you can do lots of things. Right? Okay. So as a conclusion, apa ni? Okay. Right, so that's all. So we are going to see each other again next week. Alright, so in the laboratory. So I hope we can do the, not really on the experiment and so on, but instead of, dia bukan betul real experiment lah. You just practice how to do that. Okay, any question before we dismiss the class? I think we don't have to do the class on Monday because I'm able to finish all the topics. PCR and sequencing. Okay, so then we continue only on the practical day. On the practical day, you can we can discuss on the topic as well, on the theory, and also we look at the practical session as well. We've got 3 hours kan? So I think 3 hours, so see you soon on the testing. First meet. Pakai baju raya? Tapi tak boleh bawa kuih raya, bukan dalam lab. Jangan lupa ya, Wan Muhammad Daud pakai baju Melayu. Chang Joyan pakai apa? Baju Melayu. Rini pakai baju kurung. Rini ada baju kurung tak? Duit raya dah okey. Duit raya. Okay, alright, so with that, thank you so much with your attention, with your participation, and then with your, with your time. So I hope you learn many things today, and then you appreciate the knowledge, and then the good things come from our creator, the bad things come from me, or the mistake come from me. Please let me know if you find out that there's a mistake that I wrongly tell you, so please let me know. Kalau sometimes I do mistake kan, macam baru ni dekat first year tu, totally wrong, okay, tak sedar, okay, so please let me know, just let me know, kan, boleh saja. So with that, thank you so much and have a good day. Enjoy your Hari Raya, still Hari Raya, Rayaman, Syawalman, okay. So we are going to see each other again next Thursday. Bye-bye. Assalamualaikum.
Thank you, Doctor.
Bye. Thank you.
Thank you very much, Doctor.